Okay, let's dive right into this. Welcome to this explainer. Our goal today is to demystify the absolute magic that is happening inside your cells right at this very moment. We're going to take a journey down into the microscopic world to answer a massive question. How exactly does your body take a simple set of chemical instructions and turn them into, well, you? Think of your cell as a bustling, incredibly efficient microscopic manufacturing plant. Today, we're taking a grand tour of this information factory to decode what scientists call the central dogma. Let's get started.
Here is our agenda, which is basically a step-by-step visual map of our factory tour. We'll start at the very top in the boss's office with the blueprint of life, move on to copying the recipe, and then editing the message. From there, we'll look at cracking the genetic code before heading down to the assembly line for building proteins. Finally, we'll wrap up by looking at nature's exceptions in breaking the dogma.
All right, Section 1, the blueprint of life. Let's start by setting up the grand mystery of biology here.
It really is a profound question, isn't it? How does a single microscopic cell know how to build a human, or a giant redwood tree, or a jellyfish? Every single physical trait you have, from the color of your eyes to the enzymes digesting your breakfast right now, requires specific physical parts called proteins. But, proteins don't just spontaneously assemble out of nowhere. There has to be an instruction manual.
And that brings us to the absolute foundational rule of our biological factory, the central dogma of molecular biology. Formulated by Francis Crick in 1958. Yeah, the same Francis Crick who helped discover the double helix structure of DNA. This theory states that genetic information flows in one direction, from DNA to RNA to protein. In our factory analogy, DNA is the irreplaceable master blueprint. It holds all the information, but it doesn't do the actual building. It just provides the ultimate sequence, the precise instructions that dictate exactly which building blocks to string together.
Moving right along to Section 2, copying the recipe, also known as transcription. Now, right out of the gate, the cell faces a massive logistical problem. You see, we have a master blueprint, but we can't just hand it out to the workers on the factory floor. The master blueprint, your DNA, is way too valuable to leave the boss's office, which in the cell is a highly secure vault called the nucleus. If the DNA gets damaged, well, the whole factory is ruined. So the cell uses a specialized worker machine, an enzyme called RNA polymerase, to make a safe, portable photocopy. This temporary copy is called messenger RNA, or mRNA for short. It's essentially a transcript of a specific gene that can safely travel out of the nucleus and onto the noisy, busy factory floor. This exact process is called transcription, rewriting the DNA sequence into a portable mRNA format. Now, it's absolutely crucial to understand that at this stage, we haven't actually built anything physical yet. We've merely transcribed the information from one medium, DNA, into another similar medium, RNA. We've just copied the instructions.
Which brings us to Section 3, editing the message. We've got our photocopy, but it's not quite ready for prime time.
Let's look at how the cell edits this raw transcript into our final, usable instructions. Think of the raw mRNA transcript like a newly printed stack of book pages. Before you sell the book, you really need to bind it and check for errors, right? First, the cell adds a 5' cap. Think of this as the front cover of the book. It protects the transcript and helps the factory workers grab onto it later. Then it adds a poly A tail. That's the back cover, providing extra stability. But step 2 is literally the most fascinating. RNA splicing. The raw transcript actually contains a bunch of blank or nonsense pages called introns. A tiny, incredible cellular machine called the spliceosome acts like microscopic scissors, cutting out these junk intron pages and pasting the good chapters, called exons, back together.
But wait, hold on a second. This seems incredibly inefficient. If introns are just junk sequences that get cut out anyway, why on earth does the cell bother keeping them in the master blueprint at all? Why not just have a streamlined instruction manual from the very start?
The answer is a brilliant evolutionary trick called alternative splicing. Because the manual is broken up by those blank intron pages, the spliceosome can actually choose to cut and paste the good exons together in different combinations. Imagine having an instruction manual that, depending on which chapters you skip, tells you how to build either a bicycle, a scooter, or a unicycle. Through alternative splicing, your cells can sneakily encode many different protein machines from the exact same initial gene sequence. It's a total masterpiece of biological efficiency.
Okay, let's jump into Section 4. Cracking the genetic code. Our edited message is out on the factory floor, and we need to decode it. To translate the RNA text into physical protein parts, the cell reads the mRNA sequence in a very specific way, the triplet code. The mRNA letters, A, U, G, and C, are read in groups of 3, almost like a 3-letter barcode. These barcodes are called codons. Because there are 4 possible letters, reading them in groups of 3 gives us 64 possible combinations. For example, the codon AUG means start building here, while UUU codes for a specific building block called phenylalanine. Finally, there are 3 specific codons that act as a strict stop signal, telling the factory the part is finished.
What's truly mind-blowing about this genetic code are its properties. First off, it is almost entirely universal. Whether you are a human, a mushroom, or a tiny bacterium, your cells use this exact same dictionary to translate RNA into proteins. Second, it's highly redundant, what scientists call degenerate. This just means that since we have 64 barcodes but only 20 amino acid building blocks, multiple barcodes can code for the exact same block. And finally, it's non-overlapping. The sequence is read in strict, separate groups of 3, ticking along like a metronome.
Now, because the code has multiple barcodes for the same building block, the cell developed a brilliant shortcut known as the wobble hypothesis. Often, if the first 2 letters of the codon are a perfect match, the third letter can be a bit flexible or, wobble. This means the cellular delivery trucks don't need a perfectly matching reader for all 64 individual barcodes. A single truck can recognize a couple of different similar barcodes to drop off the exact same part. It just makes translation so much faster and more efficient. But what happens when the factory gets a blueprint with a typo? Well, these are mutations. Let's look at this recipe analogy to make sense of it. A silent mutation is like writing flower like a daisy instead of baking flour. It's a typo for sure, but thanks to that wobble flexibility we just talked about, the cell still knows what you mean, and the protein is totally fine. A missense mutation is worse. It's like reading salt instead of sugar. The cell puts in the wrong amino acid, and the final protein might behave strangely. But a nonsense mutation, that changes a normal code into a stop codon. It's like a recipe telling you to stop baking now, right after you've only cracked the eggs. The assembly line halts entirely, resulting in a broken, truncated protein.
So, Section 5, Building Proteins or Translation. The crucial point is, how does this decoding actually happen physically?
This physical building phase happens inside a molecular machine called a ribosome, which you can just picture as a tiny cellular 3D printer. Translation has 3 stages. First, initiation. The ribosome clamps down onto the mRNA tape, scanning along until it finds that AUG start codon. Second, elongation. The ribosome reads the tape codon by codon, linking amino acids together into a growing polypeptide chain, which is just a fancy scientific term for a raw, unfurled protein. Finally, termination. The ribosome hits a stop codon, the machinery releases the chain, and the brand new protein folds into its final functional shape. To actually make this happen, the ribosome relies on 2 major players. The first is tRNA, or transfer RNA. If the ribosome is our 3D printer, tRNAs are the specialized delivery trucks. One end of the truck reads the 3-letter mRNA barcode, and the other end carries the specific amino acid cargo. But how do the trucks get loaded? That's the job of an enzyme with a massive name, aminoacyl tRNA synthetase. Don't worry about the jargon. You can just think of this as the diligent factory worker who uses energy to ensure the absolute correct amino acid is loaded onto the correct tRNA delivery truck. Without this worker, the whole translation process would literally fall apart.
Inside the ribosome 3D printer, there's this incredible microscopic conveyor belt with 3 stations, A, P, and E. Visualize it this way. A fully loaded tRNA delivery truck arrives at the A site, matching its reader to the mRNA barcode. The conveyor belt shifts, and the truck moves to the P site. Here, it drops its cargo, forging a chemical bond to link its amino acid onto the growing protein chain. Finally, it shifts to the E site, the exit, where the now empty truck drives away to get reloaded. This happens over and over, incredibly fast. Building your protein's amino acid by amino acid, it's just amazing.
Finally, Section 6, Breaking the Dogma. For decades, the simplified DNA to RNA to protein flow was taught as an unbreakable law, but nature is rarely that simple.
There is actually a massive historical misconception here. Take a look at Francis Crick's own diagram from a 1970 paper written specifically to correct people who misunderstood him. People thought he said information only ever goes one way.
But notice the dotted lines on the diagram? Crick knew information transfer was complex. His actual true rule was simply this: once information has been translated into a physical protein, it cannot flow out of that protein back into DNA or RNA. The physical sequence of a protein can't be read backward to rewrite your genetics. But between RNA and DNA, things get wild.
Here is exactly how nature breaks the simplified, high school version of the dogma. First, reverse transcription. Certain viruses, like HIV, carry their blueprints as RNA and literally rewrite it backward into the host's DNA. Second, RNA replication. Some viruses completely bypass DNA, just copying RNA straight into more RNA. And most shockingly, prions. Prions are infectious proteins that cause devastating conditions like Creutzfeldt-Jakob disease. They are proteins that somehow manage to replicate and spread their misfolded shape without using DNA or RNA intermediates at all. No blueprints, just spreading by contact.
Which leaves us with this final thought. We started with a neat, tidy factory flowing perfectly from the boss's DNA down to the protein assembly line. But if a protein like a prion can bend the rules and replicate without DNA, what else might nature be hiding from our textbooks? The central dogma gives us the foundational logic of biology, for sure, but it's the exceptions that remind us just how much of the microscopic world is still out there waiting to be decoded. Thanks for joining me on this tour of the cell.